So now we're going to move forward to gene mapping, which we call the 3-point cross in lab. It's basically you are looking at 3 X-linked recessive mutations. So 3 X-linked recessive mutations at once. Okay, and it has 2 main purposes, us doing this kind of a cross. The first one is it allows us to determine the order of those multiple genes that are on the X chromosome. So the first purpose allows us to determine the order of the linked genes. And then the second purpose, in addition to figuring out the order, which one comes first, which one's in the middle, which one's at the end, it also allows us to calculate the distances between the linked genes. So that's why we call it gene mapping, because in the end of a problem like the 3-point cross, you're able to map out exactly where the genes are and how far apart they are from each other. Now, the last thing I want to point out is the phenotype that we use for this cross, and that is our lovely little mutant right there on the slide. And so we use what's called the yellow, white, mini fly, which you saw already in the first lab video that I posted. So keep in mind the way we'll tend to write out this phenotype is YWM. Okay, so don't get confused throughout the 3-point cross lesson. It does get a little weird that we use the letters for the phenotype and also for the genotype at the same time throughout that cross. Okay, it'll become a little clearer in the next slides as we draw everything out. So now we're going to start drawing out what the 3-point cross concept actually means step by step. So the first thing that we would do is we would figure out what our P1 looks like. So normally we would cross a homozygous mutant female. So I'm going to say homozygous mutant female with a wild type male. And now, since we're talking about 3 genes that are linked together in a row on the X chromosomes, we're going to draw this out a little differently. We're going to draw out the genotype like this, where we draw out both chromosomes, both X chromosomes. Since she's homozygous mutant, that means both have yellow, white, mini on each of those chromosomes. Okay? Both of those are the X chromosomes. The male, on the other hand, what do you know about the male flies? Well, they only have 1 X chromosome. The Y chromosome, remember, we said is a little tiny nub of a chromosome, and it doesn't have the X chromosome genes on it. So since that male looks wild type, that tells us that on the X chromosome, each of these genes is a plus mark wild type allele. Okay? We then would cross those flies together, and when we cross them, you have to ask, well, what gametes would each of these flies make? Well, the female, she only has 1 type of X chromosome. Both of her X chromosomes, in this case, look the same. So the only gamete that she can make is an X with all 3 mutations present. Okay, that's her only gamete. Whereas the male, notice he can make two different gametes. He can pass down to his offspring either an X chromosome that has plus, 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 or what else can he pass on? He can pass on a Y chromosome. Okay, so he can give either or. So if he gives his offspring an X chromosome, well, that would make female, right? That would make the female offspring. And if he gives a Y chromosome, that would make the male offspring. And any of those offspring that they're making, that is now the F1 generation. So what do you notice the female F1 look like? Well, they got the fully mutant X chromosome from mom. Sorry, that should be W. And what does their second chromosome look like? Well, those females got a fully wild type chromosome from their dad. And then what do the male F1s look like? Well, they got the fully mutant X chromosome from mom. And what did they get from dad? The Y chromosome, which is why they are male. Okay. If you are uncertain of how we got that F1 generation, just send me a message in the Remind app, and I'll go through it step by step a little clearer. Now, when we try to figure out the F2, it's not as easy as it usually is, because with X chromosomes, if an organism has two X chromosomes, they're capable in some cases of having what's called a crossing over event when they're trying to make gametes. Now, what organisms of the flies have two X chromosomes? That's the female parent, right? So when a female has the heterozygous genotype like we saw in our F1, so I'll put a little reminder of what the F1 generation looks like. We said the females are yellow, white, mini, plus, plus, plus. They're heterozygous, while the male, F1, is yellow, white, mini, with the little y, okay? And so now, when we go to do our cross, you're going to notice that there's a possibility for this female, when she made her gametes, that she might have had some crossing over between her two X's. And what that means is the two X chromosomes, when they're near each other, they can wrap their little arms around each other and swap some of the genes. Okay, so we're going to draw out the different possibilities of what can happen with a 3-point cross with that F1. The first possibility is if no crossing over occurs. And I'll specify when the F1 female makes gametes. Well, if no crossing over occurred, well, then we have to ask ourselves, what did our female F1, what did her genotype look like? She had Y, WM, and she had a plus, plus, plus. Now she goes to make her gametes. You know what? I'm gonna erase this to move it down a little bit so that I don't end up overlapping with what I wrote above it. So again, the female, the F1 female, we just wrote as yellow, white, mini X and a plus, plus, plus X. Now she goes to make her gametes and give them to the F2 offspring. What those gametes look like depends on, what those F2 offspring look like depends on what the male F1 is giving. Now the F1 male gamete can either carry his X chromosome, which is yellow, white, mini, or the male gamete can carry that little Y chromosome to give to the F2. And the F2 are going to be in the center of this little Punnett square that we're basically making. Okay, and I'll even close it off so you get the feeling of a Punnett square. That F1 female can either give her offspring her yellow, white, mini X, or she can give her offspring her fully wild type X. And then what they get from dad never changes, right? Dad can either give an X or a Y. So if dad gives an X, that's yellow, white, mini, yellow, white, mini. Or if dad gives a Y, we draw that little nub of a chromosome. Okay, so you'll notice that these offspring, these F2 offspring, are now male. They have the Y chromosome, and these two are now female. Okay, now when you look at these offspring, you then want to ask yourself, what phenotype would they have? Okay, what phenotype? Okay, well, these two here, whether they're male or they're female, what do you notice? There's not a plus mark in sight. There's no wild type. So their phenotype would be fully mutant, right? So that's yellow, white, mini, which you'll see later on. We just write yellow, white, mini as the phenotype as well. Okay? Whereas these offspring, both of them, for each trait, you notice there's at least one plus mark. Okay? Same thing over here. Each trait, at least one plus mark. So these are fully wild type. Okay, so we'll say wild type. Basically, you would see red eyes, you'd see a normal body, and you'd see nice long wings. Okay? Now, sometimes what happens is a rare event called crossing over. So sometimes what happens on this slide, we're going to go through single crossover events. Meaning, sometimes what happens is when that F1 female, sorry, my pen just stopped working for a minute. When that F1 female goes to make her gametes, okay, normally you have her with yellow, white, mini, plus, plus, plus. Sometimes those X chromosomes cross over. So now you'll have yellow plus plus as one of her X chromosomes, and you'll have plus WM as another chromosome. Okay, because they're at this point here between the yellow and the white genes, all of a sudden the X chromosomes wrapped their little arms around each other and they swapped the end that has the first gene that we were looking at, the yellow trait gene. Okay, so now when you look at that single crossover event, well, that affects what gametes she'll now give to her offspring. Whereas the male parent, what he's giving to the offspring will not change. The male gamete will always either be his yellow, white, mini X or his little Y chromosome. Now the female, what does her first X chromosome look like? because of crossing over, the first one is now yellow, wild type, wild type. Yellow plus plus. Okay? What is her other possible X chromosome? Well, now that is plus, but where does it go? Up here, white mini. Okay, so you see that the first genes swapped places. Okay, so we're going to fill in the Punnett square as the mom giving either yellow plus plus or plus white mini. Those are her two X options. What the male gives to those offspring is the same as we saw previously. He either gives a yellow white mini X, or he gives that little nub of a Y. Okay? And then we want to take a look and see what phenotypes these F2 offspring have. Okay? So when you look at these flies, the males and the females of these F2, whenever you're looking at a set, they're going to have the same phenotype. So if you look at this female first, what do you notice? For her first gene, do you see any plus marks? No, she's got two yellow alleles, right, for the first trait. Same with the male. First trait, do you see any plus marks? No, yellow. So these offspring will have a yellow phenotype. And then when you look at the other two traits, what do you notice? There's a plus mark. There's a plus mark for that fly. So it's wild type for the rest of the traits. So we don't even need to write that because remember, we name our flies based on their mutation. Okay, so as soon as you see a mutation, that's the name. There's no other mutation there. The rest are plus marks. So that phenotype is yellow. And these F2 flies, what do you notice? The first trait of the females and the males have a wild type mark. Okay, but what do you notice about the other two traits? No plus marks around. So these flies would be called white mini flies. Okay, if you have any questions about that, you're a little confused because these Punnett squares look a little scarier than you're usually used to doing, then just let me know. I'll tell you now that I'm not going to question you on this part where we're drawing out these crazy Punnett squares. I'm going to question you on quizzes and finals the way that you'll see the final part of three-point crosses go in the upcoming slides, okay? This is just background to help you understand what we're ultimately getting to. So now possibility number 3. That female could have had a single crossover event, but on the other end of her X chromosomes. Okay, so we're going to draw this out. The F1 female again. Usually we said yellow, white, mini, plus, plus, plus. But what happens if she has a single crossover, but instead of being at the beginning of her X chromosome, all of a sudden it's at the end, okay? So now instead of the first genes crossing or swapping between the X's, now it's the last two genes that have swapped places, okay? Again, the males do not have crossing over, so in our little drawings, every time the male gametes are going to stay the same, okay? So now when we go to make our little Punnett squares, what do you notice? What's the first possible gamete? Yellow, white, and then what happened? Boom, crosses over. So yellow, white, plus from mom. What's her second possible gamete that she could give to offspring? Well, plus, plus, boom, crossover, made that last one, instead of a plus mark, it's now a mini mark, okay? So female can make yellow, white, plus, or an X that's plus, plus, M. What can the male give to the F2? Well, his X always looks the same, yellow, white, mini. yellow, white, mini, and his Y always looks the same. Okay? So these are the possible offspring if there's been a single crossover event, but at the other end of the X chromosome. Okay? And now we ask ourselves, well, what would the phenotypes look like? Well, let's look at this one. You have yellow, white, and then what's that last one? Wild type. Do we write the wild type once we've seen any mutations? No, these would just be yellow, white flies. And what about the next one? Wild type, boom, there's our mutation, so we name it after the mutation. These would just look mini, okay? Now we get to the final possibility, and this one we call the double crossover. Now keep in mind the double crossover is going to be the rarest of events to happen because the chances of a single crossover happening are pretty low. So the chances of you having more than one crossover happen when you're a female making gametes is really, really low. Okay? The way I always explain it is, well, the chances of having one celebrity visit campus one day, it's rare, but it happens. You know, we've had like Chris Rock or Val Kilmer come to campus at CSI. It happens. The chances of two of them coming to campus the same day, really, really low. Okay, but it can happen. It's possible, but it's very, very rare. So what the double crossover is going to look like. So we're looking at our female F1 fly making gametes again. Okay. Now, like we keep mentioning, normally she would have yellow, white, mini, plus, plus, plus as her two X's. But now with a double crossover event, you have the first gene, then boom, she had a crossover event, swapped with the other X, okay? And then boom, a second crossover event, swapped with the other X, okay? So this here represents 1, 2 crossover events. So that's double crossover. So each time you see that X mark, that's a crossover, not like when they have TV show crossovers. Like if you watch Arrow or Legends, any of those. So crossover events, here you have 2 crossing over events occurring. So the beginning portion of the X chromosome and the end portion of the X chromosome have wrapped their little arms together and swapped ends. Now what you'll notice is by having these 2 crossover events swapping the end genes, the end alleles, that's the same thing as if you swap out just the middle, right? If you picture it, normally I have students do this in the classroom where you line up and you swap the first 2 people and you swap the last 2 people. And when you do that, you notice that it's the equivalent of had you just swapped the person in the middle where everyone ends up. Okay? So 2 crossovers is the same thing visually as if you swapped the middle gene. The reason I point this out is that's going to get a little important, or a lot important, when we try to do actual problems with 3-point crosses where we look for order and distances. So that's going to be an important point here. Same as swapping the middle gene. Okay. Whoops, sorry. It's just taking a second because my pen stopped working. Okay, so same as swapping the middle gene. Now, we're going to do our last of our crazy Punnett squares. I know you're going to need a break after this. It's a lot, the 3-point cross. But again, it's going to get simpler when I show you how to tackle the actual problems. This is just for, you to have background understanding. So again, that male fly can either give yellow, white, mini, or little tiny Y chromosome. So let's look at the mom. What's the first gamete? What's the first X chromosome that she could give to her offspring in this scenario? Well, yellow, and then what happens? Plus M. Yellow plus M. Okay? Yellow plus M. What's her second possible? Sorry, the pen keeps dying on me. Okay, sorry, I had to fix the pen. Okay, so you have yellow plus mini. And then what's the second possible gamete she can give? Her other X now, after double crossover, looks like plus W plus W plus. And then again, you just keep filling out the Punnett squares the same way. Yellow, white, mini from father, yellow, white, mini from father, Y from father. Okay? And now we look at our phenotypes. Sorry, pen is acting up a lot today. Okay, so sorry, the pen died, so I'm going to have to use my fingers, so it might get a little sloppier. So we're going to look at the phenotypes for these flies. Again, you're just looking, do you see a plus mark for the first trait? No, you do not, so that one is yellow. What about the next trait? Yes, you do see a plus mark, so that's wild type. We don't mention it. And then we go to the next trait, which you see is mini. Okay? Then we look at the other flies, and that one is, what do you notice? Just white-eyed. Okay? So sometimes now when we do a 3-point cross and we want to look at all of the different traits, sometimes to make it a little easier for students to visualize, we'll write the phenotypes just the way you've seen the male genotypes here in the crosses. So sometimes you'll see in problems we abbreviate this phenotype as Y plus M, or this one as plus W plus, because when you have to write out 8 different phenotypes, it kind of makes it a little easier for you to visualize. Okay, you'll understand what I'm saying in a minute when we start going over the practice problems. Okay, so now we're going to work a little bit with the 3-point cross, kind of to give you an example of some of the information we can gain from looking at a 3-point cross and the results that we have. So when you look at a 3-point cross, notice now I'm going to give you an example in which 10,000 offspring were produced from the F1 flies. And you can kind of figure out some information based on the number of different flies you see for each of the phenotypes. Now notice there are 8 different possible phenotypes based on the work that we just did in the previous slide. You notice you have the 2 phenotypes when there was no crossover, the 2 phenotypes when there was a single crossover at the beginning of the X chromosomes, 2 phenotypes for single crossover at the end of the X chromosomes, and then 2 phenotypes for the double crossover. Now, when you look at results of offspring from a 3-point cross, you're actually able to figure out which of the phenotypes represented the no crossover event, the double crossover, or either of the single. So I'm going to show you how to do that now. Let's say we had 10,000 flies born from these parents that we've been working with this whole time. And notice I wrote out here how many out of that 10,000 had each of the phenotypes that you see to the left. Okay, so 4,685 were yellow, white, mini offspring, 4,759 were wild type, and so forth. Now, when you look at these numbers, you'll notice that these first two numbers over here, that's a whole lot of those F2 flies, right? That's 944 flies out of the 10,000 offspring born. That means that these two phenotypes were 94.44% of all of the offspring born from that cross. So now when you look at that, that tells you that that's a big chunk of those offspring. Now that makes sense, because what did we say about no crossovers? Well, crossovers are rare events. So for you to have a crossover is rare. It's more likely that when these flies go to make their gametes and fertilize those gametes, usually there's no crossover and they look just like the parents did, because there's been no shuffling of any genes, okay? So when you look at results from a 3-point cross, when you look at the two phenotypes that have the largest number of offspring represented, that tells you right off the bat that these were the my pen has been acting up quite a bit today, so I apologize when that happens on the screen. These are the parental phenotypes, okay? So the yellow, white, mini, and the wild type we would call the parental phenotypes. And you know that because when you look at all 8 of these numbers, all 8, let me use the laser pointer, all 8 of these numbers here, you notice that these two are most common, which tells you that they are the parental when there was no crossover. Similar to that, when you look at the other side of the spectrum, when you look at these two little numbers here, that's only 6 flies out of the 10,000 offspring, right? That's 0.06% of the F2 offspring have these two phenotypes here. Well, that tells you right off the bat that those must be the double crossovers, right? Because double crossovers are so rare. Just having a single crossover is rare. So having 2 is really, really rare. So when you have that information, you can quickly identify the parental phenotypes and the double crossover. So the parental phenotypes are the 2 largest numbers, and the double crossovers will have the 2 smallest numbers. Now, what did we say about double crossovers? We said that a double crossover, when you swap the ends of those 2 chromosomes, it's the equivalent of just swapping out the middle gene, right? So what you can do when you want to determine the order of the genes, what you can do is you compare the 2 largest phenotypes, which we have here. The most offspring have the yellow, white, mini, or they're fully wild type, right? So you compare that, the parent phenotypes, or parental, with crossovers. Sorry, my pen is acting up again, so I apologize. Parental with the double crossovers, which we have over here, which is the Y plus M and the plus W plus. And when you compare those, so that's the, we'll call that double, or sometimes you see it as DCO for double crossover. So to determine the order of the genes, you can just compare the parental with the double. Now, you already know the order of these genes because I told you, but this explanation is so that when we do the practice problem in a little bit, it becomes clear as to why you're doing it this way. So you compare the parental and you compare it to the double. And what do you notice when you're comparing these to these? Ask yourself, how do they look different? What letter is different? So in the parent, you see YWM. What do you see over here? Y plus M. Okay, that tells you that there's only one gene out of place. And what is it? The W. So in the double crossover phenotypes, when you see one letter out of place compared to the parent, that tells you that that is the middle gene in the order of those genes. Okay, so all you would do is you would then say that the order of those genes is yellow on the chromosome, then white, then mini. Now again, we knew this already, but this is just how we'll do the future problems. Okay? Now sometimes you'll see it becomes a little confusing if the letters don't look as, nice and organized as we have it here, but ultimately all you do is you look at your parent phenotypes, you look at the double, and you look to see which of these two doubles have one letter out of order compared to one of the parent ones, okay? Because sometimes it won't be Y plus M. You might see something different that looks more like, the bottom phenotype is one letter away from the top, okay? It'll become clearer as we do the practice problems. The next thing we're going to be able to do is determine the distances between everything. Okay, so the next thing we're going to do, now that we've figured out that the order of the genes is yellow, then a little further on down the X chromosome is white, and then eventually mini. We know the order of the genes, but now we want to figure out what is the distance between each of these genes here. So the way we're going to do that is first we will take a look at all of our numbers, and you notice that these two numbers here are the biggest of the numbers. So remember we said that those were the parent phenotypes or the parental phenotypes, okay? And now what we're going to do is we're going to put the percentage that each of these phenotypes represent out of the F2 offspring. So we already wrote that our first ones, that's 94.44% of the offspring. We wrote before that it's 0.06% is the last one. These middle ones, you would add them up, add them up, divide by the total of 10,000, and then multiply by 100 to get the percentage, which this one comes out to be 1.5%. And then over here, these last two here, you would add them up, divide by the total of 10,000, multiply by 100, and you should get 4.0%, okay? Once you have these percentages, then we're going to want to figure out the distances between each of the genes that we mentioned already. So the way you do that, so I'm going to give you a definition. To determine the distance, you want to add up, okay? Add up all detectable exchanges between the two genes. What this means is the first instance we want to find is the distance between the yellow and the white gene. Okay, so where have we seen an exchange between yellow and white genes? Well, in this first single crossover, remember there was a crossover between yellow and white, and in the double crossover, we saw a swapping between those two genes. So what that means is we'll want to add up the single crossover and the double crossovers that occurred between the yellow-white genes. okay? So the single plus the double between those particular genes. Now, when we add up those percentages, so 0.06, we get 1.56, but now instead of writing percent, we're going to write a special unit called MU. Okay, that stands for map units, okay, because you're mapping out the genes, so it's the units between those genes on the map, okay? So the distance between yellow and white, we would write that's 1.56 MU, okay? Now we want to find the distance between the white and mini genes. So we do the single crossovers and the double crossovers that involve white and mini. So notice that's this single crossover here and the last double crossover, okay? So we would add the 4% plus the double crossover of 0.6, and again, when you add them up, instead of writing percent, you simply write map units at the end, okay? And so going back to our picture up here, the distance between these two would be 4.06, and you would write map units. Okay, so now we're going to look at the way that you actually would encounter this kind of problem on a quiz or an exam. Okay, so I know a lot of that was confusing for the 3-point cross, but now it's going to get a little simpler because I'm going to give you a practice problem in the format that you usually see it on lecture and lab quizzes, exams, the final, all of that. Okay, so normally you are given all 8 of the phenotypes and the number of each phenotype observed. And you are asked to do the following. You are asked to tell us what are the 2 parental phenotypes, which ones are double crossovers, and how many of those did you observe. You then calculate the order of the genes, the distances, and ultimately draw out the full map. So I want you to pause the video now and take a minute or a few minutes to try and answer all of these questions based on the chart on this page. See if you can do that, and then move on in the video to the next slide where I then explain each of the answers, okay? But I want you to try it first on your own to see if you are clear or if you need a bit more practice. Okay, so hopefully now you try to do the problem on your own. And now we're going to go question by question to show exactly how to tackle this kind of a problem. Okay, so the very first thing you're going to want to do is you're going to look at all of the numbers representing the number of offspring of each phenotype observed. And I want you to, first thing you always do with a problem like this, circle the 2 largest numbers, okay? So 300 and 290 are larger than the rest of any of those numbers. So right away, that tells you that whatever phenotypes are there, those are the parental phenotypes, the phenotypes in which there has been no crossover event. Because remember, we said crossing over is quite rare. So most of the time, the fly will not have any crossovers, and you will see the same offspring as you saw with the parents. Okay, so parental phenotypes would then be these 2 here. You could write it as YSV and plus, plus, plus. And again, I know that's what I mentioned earlier would be a little confusing, that we're using the symbols we usually use for genes as phenotypes, but it's to make it easier because we're dealing with 3 different traits at once. Okay, so you would have the fly that looks like YSV, which in flies is yellow sepia vestigial, but you don't have to worry about that. And then the fully wild type fly. So those would be your parental phenotypes. Now keep in mind, sometimes you'll do a problem and the parental phenotypes will not be 3 mutant letters and 3 wild type. Sometimes it'll be 2 mutant letters and a wild type. or one mutant letter and two wild types. So it's not automatically the ones that have 3 mutants, 3 wild type. It's automatically the ones that have the largest numbers of offspring. The next thing you do anytime you encounter this kind of problem is you circle, or in this case I'll do a square so that it looks different for you, the 2 smallest numbers, okay? So parental phenotypes are the largest numbers observed, and then the double crossovers are the smallest numbers. Because what did we say about double crossovers? A crossover event is rare. Usually doesn't happen. Can sometimes, but it's rare. Double crossovers are even rarer, even less likely to happen. So you're only going to have a few flies that are double crossovers. So you look to your double crossovers. The double crossover phenotypes, if I asked for that, that would be YS plus and plus plus V. But what did I ask for? I asked how many. Okay, so how many, you would then do 5 plus 7, right? Okay, so you would then tell me that there were 12 double crossovers. Okay, 12 flies represent the double crossovers. Once you know your parental and your double crossovers, the highest and the smallest numbers, you can then figure out the order of the genes. The way you do that is you look at your parental and you ask yourself, does either of the double crossovers look like my parental but with one letter out of place? Well, you notice this one right here. That looks just like YSV, but with one letter out of place. It's the last letter. What letter is out of place? The V. So what does that tell you when you compare parental to double? Okay, parental being YSB and plus plus plus, double being YS plus and plus plus V. You notice the one letter that looks different between the two is V. So that tells you V is the middle letter. So the order of your genes would be, if we draw the chromosome, it would be V in the middle. And then you would place the other 2 genes around it. And it doesn't matter which you put first, which you put last, because a chromosome is 3-dimensional. All that matters is that you have the middle gene correct. Okay? So now that we know the order, the last thing that we'll want to do in order to fully map this is we'll want to know the distances between each of our genes. Okay? The way that we do this is first we want to know the distance between Y and V. So we need to add the single crossovers plus the double, right, for the Y to V. Double is easy. We already know that the double crossovers, how many did we have? We had 5 plus 7, which is 1.1% of our flies. But before we get to that, we have to figure out how many Y to V single crossovers did we have. Well, we already know now the order of our genes, right? We know the genes are Y, V, S, right? So if the genes are in order Y, V, S, then if you had a single crossover event between Y and V, what would suddenly be out of order in your parent? The Y. The Y's would suddenly be swapped. So we go to our parental phenotypes, right? Our parental phenotypes, which are over here. And we ask ourselves, if you took this parental phenotype and you swapped the Y, right, with the plus, what would you end up with? Well, you'd have plus SV, right, if there was a single crossover there. Where do you see plus SV? Over here. So that tells you that these phenotypes here represent a single crossover between Y and V, okay? And that then leaves you with these guys over here to be the single crossover between V and S. And the way you could have checked that is you'll notice that what's out of place from the parent, the S is suddenly out of place, because if you had a crossover in the back there, the S would suddenly be out of place. Okay? So we take our Y to V single crossovers, which are right here, 160 plus 168, and we turn them into a percentage by dividing by the total number of flies, which is 1,005. So we could get that by adding all of these observed up and multiplying by 100. You then add that to the percent of double cross, okay? So think of these as percentages. The percent of double cross would be 5 plus 7 double cross flies divided by the total multiplied by 100, okay? When you do that, you should end up with 32.6 plus 1.1%. But what do? We write that as map units. So the total is 33.7 map units between Y and V. So when we draw the map out between these two, you would have 33.7 map units. Now we want to do the same thing, but we want to find the distance between V and S, right? So the distance between V and S now is the single crossovers of V and S plus the double crossovers, okay? Where did we say the single crossovers of V and S are? Right here, okay? So we would have to add together 40 plus 35, turn that into a percentage by dividing by total and multiply by 100, and then you add that to the double crossovers, which we already did in the previous one, so you already know that that's 1.1. Okay, when you add these together, you get 7.5 plus 1.1, and so what does that give you? 8.6 map units between V and S. So if we drew out our final map, we would have Y, then you would go 33.7 map units away, you would get to V, and then you would go 8.6 map units away before you got to S. Okay? I know that gets to be a little confusing, but if you follow step by step, it's not that bad. Anytime you get one of these problems, the first thing you should do is look for your largest two numbers and label them as the parental phenotypes. Then look for your smallest numbers and label them as your double crossovers. Sorry, my pen died again, so I just have to switch over. So you look at your smallest numbers, and those become your double crossovers. You then compare the phenotypes of the parental and the double crossover, and you ask yourself which letter is out of place. Whichever letter looks different in the parental versus the double crossover, That is your middle letter in the order of those genes, okay? Once you have that, then all you have to do for the distances is tackle each part of that map separately. So first you do the distance between the first two genes by adding together the percentage of single crossovers with the percentage of double crossovers. Now again, please remember anytime you are calculating a percent, you add together the two numbers observed, divide by the total number of flies observed, and multiply by 100. That gives you a percent. Okay? When you then add together these percentages, keep in mind at the end, do not write percent, instead write map units. The last thing I want to recap is when you are trying to figure out which single crossover is which, okay? Write down or look at the order of your genes and ask yourself, if you have a crossover between the first two genes, what would be out of place? The first gene, right? You can look back at our notes on the big chart that we made for the example before, and you could see that the first letter would suddenly be out of place in the parent, okay? And so once you see that, for instance, in our example, the single crossover between Y and V, Y will be out of place when you compare the parent with the offspring phenotype, okay? So notice that suddenly Y is swapped. So instead of the parent, YSV, you suddenly have plus SV because Y has been swapped. That tells you right away that's the single crossover for those first two genes. And then you do the same thing with the other two genes, the N genes, or just by process of elimination. All you have to do is identify one set of single crossovers like we did, and then automatically that means the last two numbers you're left with are the other genes single crossovers, okay? If you have any questions, any concerns with how this was done, or you want me to draw out another example for you, please send me a message and remind. The best way to figure out 3-point crosses is to really slow down, review those steps that I just went over, looking at what's parental, what's double, what's the order, what's the distances, okay? Do it in that order every time. Even if the professor only asks you for, let's say, the order, do it in the same order that we just did. What's parental, what's double, then figure out the order, okay? So if you remember the steps on this page, you'll be okay. Now we get to another practice, okay? I want you to stop everything, grab your phone, and I want you to look at the chart on this page, okay? It's just like what we did in the previous slide, except I've changed the letters of the phenotypes and changed the number observed for each. In lecture and in lab, you'll get a similar format when you do 3-point questions where you will be given a chart, you'll be given 8 phenotypes and 8 different number observed, okay? What I want you to do is right now, when you get to this slide, pause the video and send me in Remind app what you think the order of these 3 genes would be, and tell me why you think that's the order, okay? If you would then like to give me all of the other answers that we just did for extra practice to make sure you really understand it, then you can, in addition to telling me the order, you can tell me what the parental phenotypes are, what the double crossover are, what the distances are, okay? When you do this, please also send me a picture of this slide so that I don't have to look back in my notes to remind me the exact numbers that I use, because each time I give the class slightly different numbers. So if you could just stop everything, send me a picture of this slide, and tell me your answer as to the order and as to anything else that you would like to go over with this practice problem. Okay? That is it. That's it for today. When I post this as videos, I'm going to post it as 2 halves because I know it's a lot of information. So if you want, could have gone through just X-linked and then 3-point. Now, if you have any questions, you need a little help with any of the topics, whether it's the X-linked crosses or practicing the 3-point map, sometimes it takes a few times looking at it before all of a sudden it will click. Just let me know. Contact me in Remind, and we can kind of go over these topics in a little more detail. And again, whenever you send me anything in Remind, if you could also send me a picture of whichever slide you're asking about, that makes it a little easier for me to quickly respond to you without me having to double check exactly what part of the lesson you're referring to. Okay? Thank you and have a great day.